Hello there, everybody. We are now on to the second part of our molecular genetics section. And this is going to be on something called DNA transcription translation. And it is something that's valid to think about. We talked about a process called mitosis, right? And in mitosis, you have cells that duplicate themselves constantly. So these cells are constantly duplicating themselves. We call them actively mitotic cells. And we talked about reasons why cells need to become mitotic. And one of the reasons why is that you have on the bottom layer of your skin, you have these cells called stem cells. And these stem cells are constantly dividing, like constantly. They're constantly dividing, and they're what we call undifferentiated. So what happens is that as these cells are made, as they keep on dividing via mitosis, eventually they begin to express certain genes to make a protein called keratin. So what's happening is that the stem cells are using information in their DNA in order to make proteins needed to become skin cells, which is why that every time you develop a new top layer of epidermis, it looks exactly the same. It's because simply is that these stem cells are differentiating themselves into skin cells called keratinocytes because they're reading parts of their DNA that contain the proper genes for how to make the protein keratin. They're following the instructions contained in those genes, and they are actually building the proteins needed in order to create mature skin. And these are examples of what we call proteins. So we're going to start by talking about proteins. And what proteins are is that they are comprised of chains of amino acids. If you remember one of our first lectures, we talked about biomolecules, and amino acids are the biomolecules that comprise proteins. So what happens is it's very interesting. We actually have 20 odd amino acids. Let me go ahead and show them to you. So amino acids list. Okay, so I'm going to go ahead and show you this here. These are the characteristics of all the amino acids that make up proteins. And you're seeing here that they have different characteristics. Some are nonpolar, some have a positive charge, some have a negative charge, some are polar. If you take a more advanced biology class, you'll learn exactly what these things mean. But these amino acids, what they do is that they're placed in a different order and in different quantities. And what happens is that when the protein is built, it's going to fold into a unique structure. So these are the biomolecules known as amino acids. We see that there are several different types. I believe there's 20 something. And these amino acids, they're actually stored in your cells. So right now, there's a bunch of amino acids just floating around in your cells, specifically near the rough endoplasmic reticulum, but they are here. Now what happens is that those building blocks, as I said, the amino acids are there. The question, of course, is what order do the amino acids have to be in to make the correct protein? The correct protein. So here is the analogy. we're going to say that the gene is going to be a recipe for chocolate chip cookies. I don't know why I use chocolate chip cookies as the example. I don't even like chocolate chip cookies. But for whatever reason, it's my go-to. Like, every time I go on a diet to trim down a bit, I always crave pizza. I crave pizza. I dream about pizza. I don't even like pizza. The one time I gave in when I was dieting and had a pizza, I didn't even like it. I'm like, what am I doing eating this thing? I don't even like it. The same thing with chocolate chip cookies. There's probably my least favorite type of cookie, and here I am using it in every example. I digress, though. Just sometimes you just have to fast forward some sections of my lecture. So if the gene is the recipe for chocolate chip cookies, the amino acids are the raw ingredients. Now you have these ingredients in your cupboard, just kind of hanging out there. However, the final product, I'll just say the baked good is cookies created by different ingredients and quantities. So that's similar to building a protein. The gene is wait, why did I write baked good? I meant protein. What am I doing? So the protein. All right, yes. So anyways, the gene is located in the DNA. Recall that DNA segregates into chromosomes. Chromosomes have different genes. The amino acids are located in the rough ER. And the protein is made in the rough ER and stored in the Golgi. Okay. So this is what I mean by a protein. This is actually something that's pretty neat if you want to try it out. So we're going to go back to our screen here. And I'm going to go to the NCBI database. I think I told you guys about this before, but it's the website that is used for geneticists to look up any gene sequence, any DNA sequence that they need. And probably the simplest protein, or the one of the most researched, is called HBB. Stands for hemoglobin beta subunit. It's one of the proteins that makes up hemoglobin in red blood cells, also known as erythrocytes. That is their more proper name. Hemoglobin beta subunit in Homo sapiens, that's humans. So I'm going to go ahead and look up this. Okay. Yes, it is. Every gene for every species has its own page, its own unique page. Think of it like a Facebook for the gene, just not nearly as interactive, no friends list. Not exactly the most user-friendly. But anyways, what we are looking for here is this thing here, this sequence here. This is the gene sequence for hemoglobin. You'll see that it's different combinations of A, C's, G's, and T's. Because recall that genes are going to be DNA code. Okay, and so we're also going to see here that says exon. We'll talk about that in a bit, but if I click on CDS down here, you're going to see something interesting. Out of this gene sequence, which is 601 nucleotides long, only only about maybe 3/4 of it is actually highlighted in brown. This is because DNA has a very interesting characteristic, is that some of the coding, actually the majority of the coding, is not actually used as instructions for making the protein. Think of it as when you Google a recipe online, how there's all this background information. You're like, okay, whatever. And then you just skip to the part with the recipe. That's similar to how genes work, is there's a part of the gene that doesn't code for anything. It's there, but then when it's time for the protein to be made, when it's time for the gene to go to the rough ER, part of it's just cut out. All the stuff that is not highlighted in brown is cut out. We call them intronic regions. So anyways, what's going to happen here is that this sequence here in brown, this sequence is going to correspond to different amino acids, which are here. This is called a translation. Each of these stands for a different amino acid. So if we look here, ATG is going to correspond with the amino acid methionine. GTC is going to correspond to the amino acid valine, so on and so forth. CAT, you remember this one from our talk on mutations. CAT is going to code for histine. And then as soon as this chain of amino acids are strung together, it's going to fold into a protein. And if you want to see what that looks like, you can go to a website called SwissProt. And this website is super awesome because it actually uses computers to build proteins. Here we go. So what you do, it's very cool, if I can find it. As I said, these websites are not exactly user intuitive, to put it nicely. No, as in they don't want you to use it unless you are absolutely looking for it. I want Swiss model. Where is it? Swiss model. Where are you? Okay, it's being very elusive. I'll look up Swiss model. I hope they didn't take it offline. That would make me sad. My Friday nights will never be the same. Ah, here we go. Good. Friday night saved. So what you do is you can actually paste in the sequence of amino acids using the first letter as the key, and you click build model. It's going to take a while because it's actually building your protein. And when we finally get it, it's going to look not like you would think. We can come back and check back later. But basically, what's going to happen, and I'll show you this later, is that when the amino acids are strung together, it's going to fold into a shape depending on the combination of the amino acids combined. It's going to form into a shape, which is why it's very important that the gene sequence is not changed, because if it is, then it can significantly change the protein. Like how I was talking about, if there's a mistake in a chocolate chip cookie recipe where sugar is substituted for salt, yeah, it's only one change, but it's going to drastically change the final product. Very similar with proteins, is that if just one mutation happens in a gene, it can significantly change the protein to the point where it's not even functional. All right, so we know about proteins, we know about genes. Genes are sequences of DNA that code for a protein. There's another term called gene expression. So anyways, gene expression, what this is how often the gene is transcribed and translated. We'll talk about those terms in a bit. So how often the gene is transcribed and translated. If we say that a gene is expressed often, that means it's read and transcribed and translated fairly often. So the protein is made often. If the gene is not expressed often, that means that it's not turned into a protein very often. Think of gene expression as analogous as how often you cook something. If you cook something a lot, you express the recipe often, and if you don't, then, well, you don't express it often. Now we have something called a regulatory sequence. So genes have two aspects to them. One is called a regulatory sequence, which determines how much protein is made. And then we have the coding sequence. The coding sequence determines how to make it. So genes have two sequences. The regulatory sequence controls how much and how often. The coding sequence states how. What they'll do sometimes is if a gene is expressed very often, what they can do is they can create a hybrid gene where they take the regulatory sequence of one gene and add it to the coding sequence of another. So what that can do is take a gene that isn't expressed very often and make it highly expressed, and vice versa. All right, so now we're going to talk about transcription and translation. So I'm going to draw here a cell. And in the cell, we know that there is DNA. I'm going to draw the nucleus rather large. We have DNA. Remember, it is a double helix structure. Then outside the nucleus, we have the rough endoplasmic reticulum, which is the site of protein synthesis with the ribosomes. Here's the thing. The DNA never leaves the nucleus. It is there forever. We also know that the DNA is analogous to a recipe book that is like 5,000 recipes or more and weighs like 400 pounds. So it's not going anywhere. It makes sense that if you want to make a recipe, you just copy that page and just walk the recipe back home. Well, that's exactly how protein synthesis happens. Is a copy is made of just a tiny, tiny, tiny part of the DNA, and this copy is transcribed on a piece of genetic material called mRNA. We'll talk about RNA in a moment. This messenger RNA is going to leave the nucleus and it's going to go to the rough ER, where it's going to be read and translated into a protein. So translation happens on the rough ER, and transcription happens in the nucleus. So the goal is we want to take just one gene contained in DNA, more or less, in a sense, copy that information onto the mRNA, where it's then going to go to the rough ER and it's going to be turned into a protein. Translation. So let's go ahead and talk about RNA. RNA was the very first, we think, the very first genetic material that was created on Earth. It's simpler. It's simpler. For one, the sugar is ribose instead of deoxyribose, which for the intents and purposes of this course means nothing. It has a similar structure as DNA, but it is single-stranded. And as far as its 4 bases go, well, there's a slight difference there. So I'm going to draw just a example of what one looks like. So it follows the same rules as DNA, almost. The issue is that RNA has a couple of characteristics. One, it is single-stranded. It is much smaller because it only contains a very small amount of genetic information. Also, it's mobile. But probably the biggest issue is that it doesn't make the base thymine. Instead, thymine is going to turn into uracil. Now, what is the difference? Well, RNA, for whatever reason, doesn't have the thymine base. Instead, it has one that's very similar called uracil. So it's not quite as good as thymine. Think of thymine as Oreos, and think of uracil as the store brand sandwich cookies. Now, they look like Oreos. They can be substituted for Oreos, but they don't taste quite the same, and they don't look quite the same, but they're basically Oreos. That's uracil. For whatever reason, RNA, a more primitive molecule, just doesn't have that advanced base thymine. But uracil is a decent substitute. Everything else is the same though. So for the intents and purposes of this course, uracil acts the same as thymine. You just have to remember to substitute it. So it still has it still has adenine, cytosine, guanine, but instead of thymine, it has uracil. So uracil is going to bind with adenine this time. It's basically the same. It just is uracil. All right, so that being said, I am going to draw here an example sequence for DNA. I'm going to write something called a sense strand. So now we are back, what the heck? Now we are back to DNA. What the heck? I just want to, there we go, sense strand. Now I'm going to write this for the sense strand. A, T, G, C, C, A, T, T, A, A, TGA. So what I'm doing is I'm basically just giving us a sample gene. And I'm going to write a second strand called the antisense strand. Antisense. And the antisense strand is going to follow the base pairing rules. So DNA is double-stranded, right? Well, one of these strands we call the sense strand, one we call the antisense strand. The antisense strand is going to follow base pairing rules. So T, A, C, G, G, T, A, T, 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 A, C, T. All right, so what is this sense strand and this antisense strand? Well, the sense strand contains the information necessary for making a protein. So it has necessary information. However, the antisense strand does not. It has nonsensical information. Nonsensical information. Adding to our example, what if the chocolate chip cookie recipe, what if the next recipe was for like concrete soda bars, and it was like add a pound of wingless beetles, put in some honey from the ancient bees of the Roman era, and you're reading it like this makes no sense. That's kind of like the sense and the antisense strands. Is the sense strand contains the information necessary for making proteins. However, we have a whole nother strand called the antisense strand that is nonsensical. You're probably thinking, well, why even have a double-stranded molecule? Why not have a single-stranded one? Ah, that's a good question. And the answer is that our genetic machinery has a quirk. And I'll write that here. DNA cannot be copied. However, this is the big however, a new strand can be synthesized using base pairing rules. That is the situation. We just can't copy DNA, but we can read one strand and build a new one using base pairing rules. Now, this has to be done by enzymes because remember, enzymes are proteins that either build things or break things down. We have an enzyme called RNA polymerase. You may remember it as being RNAP, so RNA polymerase. So what RNA polymerase is, it is an enzyme that reads the antisense strands and builds a new strand. Sorry, that's kind of messy, but it says builds. Oh gosh, that's going to bother me. I have a tolerance for messy handwriting. Builds a new one using base pairing rules. That's what it does. That's its job. Remember, enzymes have very specific jobs. They can't deviate from that because they don't have free will. Okay, we'll call a spade a spade. It just does what it's designed to do. What this RNA polymerase is going to do is it's going to go down this strand here, and it is going to synthesize a new strand using base pairing rules. It's going to read the T and it's going to put an A. It's going to read the A and it's going to put a T. No, it isn't. It's going to put a U. Because remember, RNA does not have uracil. Sorry, RNA has uracil, not thymine. So anytime you see you think, well, I need to build a thymine. No, RNA has uracil, not thymine. C goes G, C, C, A, U, U, A, U, G, A. You know you did it right if you compare this new RNA strand with the sense strands. They should be identical, except uracil should be there for thymine. And if we look here, we see that the sense strand and the new messenger RNA strand are identical, save for the U's instead of T's. Now, something's going to happen once this RNA strand is made. It's going to be modified. So what's going to happen next is that the immature RNA strand will be modified. It's going to undergo 3 modifications. The first one is that it's going to have introns excised out. So the first step is that introns will be excised out. Now you're probably saying, what is an intron? Well, I'll explain that. Introns excised out. So DNA has 2 sections to it: exonic regions and intronic regions. Let me write this over here. So introns sections of DNA that do not code for proteins. And you're probably thinking, well, how do I know if an intron, if a part of the gene is an intron? Well, that's because as soon as this RNA is made, it's going to have certain sections cut out. So we'll say that this section here, which is AAA, it just is going to be cut out. So we know that those are intronic regions. So now we are going to have our strand that's now going to have a part of it cut out. Like when I showed you the hemoglobin sequence, only a certain part was considered exonic regions. So the introns were cut out. So now we have step 1, the introns are going to be excised out. The part that remains are called the exons. And these are sections that code for proteins. I guess maybe it's similar to when you write an essay for English, and you put in a part that really is not necessary. In fact, it really serves no purpose, so you just take it out. It's similar to that. Now next, something called a poly-A tail is going to be added. Poly-A tail. Poly means many, A is for adenine. So what's going to happen is that on one end of this RNA, there's going to be a bunch of A's added. And they think this is for identification. This is so if when our cellular machinery sees this messenger RNA moving about, if it sees the poly-A tail, it means that it was made by the cell. It's also going to have something added called a GTP cap, and this is for energy. GTP cap. This is for energy for the mRNA to move. It's going to be for energy. Now, this is going to be the end of the process called transcription. So we just went through transcription where the sense strand of DNA is turned into RNA. It's called transcribed because it is written on a different format. Like if you were to take a book and read it and record it, then you would be transcribing it. These would be the same information just from one media to the other. So now this is going to go into the rough ER. Okay, let me make sure I have the sequence down. Because I'm going to definitely forget it the moment I hit erase. So let me make sure that I have this here. All right, so A, U, G, C, C, A, U, U, A, U, G, A. All right, great. What's going to happen now is that this sequence is going to leave the nucleus. I should note that this is all happening in the nucleus. It's going to leave the nucleus and it is going straight to the rough ER. Now recall the rough ER is just outside the nucleus. So here I'll draw the cell again. This is the nucleus. This is DNA. This is the rough ER. And these dots are the ribosomes. So, and now the messenger RNA is going to leave the nucleus and it's going to the rough ER. And I'm going to draw here a ribosome. It kind of looks like a sandwich. So this is the bottom part of the bun. This is the top part. And the messenger RNA kind of goes into the middle. So AUG, CCA, UUA, UGA. The reason why I'm putting them into threes is because every 3 nucleotides is called a codon. A codon is just 3 nucleotides. And why is that? Because codons refer to a specific amino acid. It doesn't matter what order they're in. Every three nucleotides is called a codon, and they all correspond to one unique amino acid. Well, actually, that's not entirely true because sometimes multiple codons refer to the same amino acid. But anyways. Okay, so this process is now going to be called translation. And translation is where the mRNA code is turned into a protein. So what's going to happen here is that in the rough ER, there are amino acids everywhere. I'm not going to get too much into this because it's going to complicate things a little bit too much. But what's going to happen is that the ribosome is going to read each of these codons. It's going to read it and it's going to say, okay, AUG, this corresponds to the amino acid called methionine. And it's going to start the amino acid chain with it, MET. Methionine is a very important amino acid because methionine is what we call the start codon. This signals to the cellular machinery that it's time to begin a new protein. Saying, okay, I see methionine, it's time to begin a new protein. It's like a start button. So methionine is a very important codon. Now, what about CCA? Well, let's go ahead and let's look at a codon table here. Oh, by the way, our protein is made. I'll show it to you afterward. Keep you guys watching. So I'm going to show you now a codon table. What's kind of neat is that this is exactly how our cellular machinery knows which amino acid corresponds with each codon. It actually follows the same exact formula. So when you take your quiz and your exam that's coming up, you will need one of these codon tables. Well, actually, I'll give it to you, but yeah. So if you look here, these are all the codons that correspond with different amino acids. So what it does is ribosome reads it and it's going to go, okay, CCA, that means I need to put in a proline. So let's go back to our whiteboard. CCA is going to correspond with proline, which is abbreviated pro. Next is going to be UUA, and UUA is going to correspond with leucine. LEU. And then UGA is going to correspond with stop. Stop is a codon that tells the cellular machinery it's time to stop making the protein. The protein is done. It's like the part of the recipe where it says, okay, let cool and serve. So the stop codon states to terminate the sequence. It is done. Terminate the sequence. Now what's going to happen here is that as soon as the sequence is terminated, this amino acid chain is going to be allowed to do something called fold. Think of it like the amino acids are going to talk to each other, and they are going to decide what shape to turn into. So it's going to start out with a long chain, and then it's going to fold in a certain way. For example, like this. So protein folding is where a newly formed protein twists into its correct shape.